Thanks so much for Oxford Nanopore to invite me here to present our recent work, that is how do we achieve the telomere-to-telomere genome assembly with nanopore simplex rate. So how do we assemble a diploid human genome? So consider we have a real diploid genome, including two haplotypes, one from the father and another from the mother. So most of the existing algorithms, actually, they traditionally can only output the collapse assembly, which is a mixture of the two haplotype and at least losing half of the information for a diploid human genome. So after that, there are several methods that are trying to reconstruct more information. So they would like to output one primary assembly and one alternative assembly. However, the primary assembly is still a collapse assembly, and alternative assembly is highly fragmented and making it hard to be used in the downstream analysis. Most of the current sequencing projects would like to output the haplotype resolve assembly, or we call the phase assembly. So their aim is to output two assemblies for each sample, one for each haplotype. So this could reconstruct most of the information about a diploid human genome. But however, the ultimate goal for the de novo genome assembly is the telomere-to-telomere genome assembly, which aims to reconstruct the entire chromosome from the end to the end. So the first T2T human genome assembly was completed in 2022. Although it is a relatively simple haplogenome, only including one set of haplotype, it still required multiple sequence technology with very deep sequence depths, and it's actually a result from the manual creation with a large team effort. So there are more and more new T2T genome assembly projects are coming out, but all of them rely on the complex hybrid assembly strategy that require multiple sequencing platform. It's quite complex and not easy to be achieved. So current strategy for the de novo assembly with long reads, they mostly trying to using three types of long read data. So the first is the peptide reads. So these reads are relatively shorter, but they are more accurate. For the nanopore sequencing platform, they could always provide two types of data. So the one is ultra-long sequencing data, which could get the read length up to the 200 megabase, while the nanopore standard reads, actually, they're relatively shorter, but still longer than the peptide reads. So the disadvantage for the nanopore sequencing technology is that they have the relatively higher sequence error rate, maybe not so much, but it still makes the current de novo assembly algorithm cannot work with the nanopore simplex reads to produce the phase assembly or the T2T assembly. So as a result, most of the current pipeline for the T2T assembly require both the peptide reads and the nanopore ultron reads. So this is quite expensive and require much more DNA. So the aim of our method here is trying to reach out to the near T2T assembly using the cost-efficient nanopore standard rates. Although these rates are shorter than the ultron rates, but they are still longer than the peptide rate. So the challenge for the de novo genome assembly with nanopore simplex rate is mostly coming from the sequence errors. So the existing haplotype resolve assemblers are not optimized for the nanopore data. They are all originally designed for the high accuracy rates with random sequence error, like the example on the left side. So the purple dot represents the sequence error, that actually a random sequence error that appears in one read. But for the nanopore simplex reads, they have more recurrent sequence error. So if we look at example on the right side, actually there are two purple dots actually within the same site. So they are recurrent within the two reads. So this makes the de novo assembly algorithm cannot directly work with the nanopore simplex reads. So the existing workflow for the simplex assembly actually requires additional deep learning-based error correction methods. So if we look at the workflow on the bottom side, actually we see that we can run the hero or the direct correct to remove this 6 error within the raw reads and then get the near error-free corrected read, and finally feed this read back to the de novo assembly algorithm. But this method actually has a issue for the computational efficiency. It requires substantial computational resource with the high ends of the GPU, large memory requirement, and a long running time. In addition, the T2D assembly currently can only be feasible with nanopore ultron rate. So as we mentioned before, this ultron sequencing has a higher sequencing cost, and also it requires higher DNA input. So what we are doing here is that we propose a new error correction method in our hifiasm ONT framework. Actually, can directly work with nanopore simplex rates. So if we look at the example on the right side, which showing a basic idea of our methods, it's trying to cluster the potential real SNP of the heterozygous alleles using the long-run defensive formation, and then discard any of the isolated sites that are more likely to be the sequence error. So eventually we can use it using some of the efficient dynamic programming method to do that. So it will make our method much faster and no GPU requirement. So we also do some optimization for the hifiasm, actually for the ONT rates. So here is an example about the correction results. So on the top track, we have the raw simplex rate, and the bottom track, we have the corrected rate after the hifiasm. We could see that nearly all of the sequence error can be removed, and there are just a few heterozygous sites remaining. That actually this is the result that we would like to have. We would like to keep the real biography effect. So here I'm showing the result about the near T2T human genome assembly with another standard rates. So we have the 7 human sample from HZ01 to HZ07. So each sample will be base calling using the super base calling model and average read length 30 kilobase. So if we look at how many telomere-to-telomere contact and scaffolding we achieved, that we could say the hifiasm is much better than the Verco plus Hera. So the hifiasm algorithm here can achieve about the tens of the telomere-to-telomere contacts, but Verco and Hera cannot achieve any of them except the HZ01 with 2 T2T contacts. So in addition, we compared with this nanopore assembly with the HiFi assembly, we could see that the assembly is also much better than the HiFi assembly, mostly because the nanopore is tens of kilobits longer than the PyBar HiFi rate. So for the running time, actually, the HiFi assembly is also nearly 10 times faster than the Verco plus Hera, and also the HiFi assembly does not require the GPU. We also test our method with nanopore ultra-long rates. So here we test the 5 human sample and the non-human sample. So as we expected, that the hifiasm also nearly 10 times faster than the Verco plus Hera, especially for the error correction part. And for the T2T chromosome numbers, the hifiasm is also better than Verco plus Hera, especially within T2T context. So especially for the human genome, you could say that the human genome actually have the 46 chromosome, but the hifiasm can achieve about the 44 or 40 or more than 40. Actually, it can make the most of the chromosome telomere to telomere. We also test our method, especially for some medical relevant genes like the SM1 and SM2. These are highly repetitive genes. We compare the assembly to the HC02Q100 reference. So this is the ground truth from the SAM sample. So we could say that for all haplotype and either data type, actually the Verco plus Hera has an issue for resolve this region. So especially for the standard nanopore rate. But for the hifiasm, it can resolve the SM1 and SM2 using either the ultra-long rate or the standard nanopore rates. So especially for the standard nanopore rate, we think it's pretty important. It's just because it required about the 40 times less DNA input than ultra-long sequencing. So this will make the de novo assembly much more feasible within the different type of application. So in summary, that we propose the hifiasm ONT algorithm that can enable the direct assembly of the nanopore simplex rates. It requires significantly fewer computational resource, and we demonstrate that this T2T assembly is feasible with the nanopore standard simplex rate. We believe it will significantly extend the application of the T2D assembly, especially for the clinical sample that the ultra-long sequencing cannot be available in most cases. So in the next step, we would like to say if we could do some of the ongoing development to further improve our algorithm, for example, for the assembly-based accuracy and also the new hybrid assembly strategies. Yeah, so we would like to thank the support and the collaboration from the Oxford Nanopore team, and also we would like to thank all the co-author and the Human Pendulum Project, and also the funding support from the NIH. I'll stop here and thanks so much for your attention.